Right. Very good evening, dear students. Put this work on slide 128. I hope we are doing fine, and today's lecture we are going to explore two lipoproteins and atherosclerosis. So our learning objectives for this lecture is to understand what lipoproteins are, we understand what atherosclerosis is, and we start the relationship between lipoproteins and atherosclerosis. I think there is really a spelling error right there. Let me try to correct it. Yes, atherosclerosis. They will also explore in length how the body makes lipids, and then we understand the regulation of lipid synthesis in the body. One, we talk about the digestion of these lipids, that during chewing it just breaks down the large lipid particles into smaller pieces. So this helps increasing surface area for further digestion. Then there's an enzyme called the lingual lipase in the mouth that is contained in the saliva. It initiates preliminary breakdown of triglycerides into fatty acids and glycerol as well, and especially for the short chain and the medium chain fats or lipids. Now, once that is done in the mouth, the next step is digestion in the stomach, and this is limited. There is a limited fat digestion in the stomach due to the acidic pH. It's all usually proteins that is digested in the stomach. There is actual mechanical form of digestion where food undergoes churning, and this helps to mix it with juices and further breaking down lipid particles. We also have the bile salts, which are released from the livers and are not released here due to the acidic environment. So the bile salts won't be released because of the acidic environment. In the small intestine, which is the duodenum and the ileum, and that's the final step for the digestion of lipids, is that we have the pancreatic lipase. These are the key player of digestion here, that they break down triglycerides into fatty acids and monoglycerides. Bile salts are then released from here, the bladder, and this basically help in emulsifying the fats, and then they create microscopic micelles for better absorption. We have the coenzyme lipase, and this aid in the pancreatic lipase. It helps pancreatic lipase to perform its function of cleaving lipid into fatty acid and monoglycerols. Absorption in the bloodstream is that the micelles that reach the absorptive surface of the small intestine, that's on the lining, and that the fatty acid and monoglycerols will diffuse into intestinal cells. Reassembly, and inside the cells, the fatty acids and monoglycerols are reassembled into triglycerides again. And these chylomicrons, which are the fast transporters, so lipids, their triglycerides are packed with cholesterol and protein, proteins forming what we call the chylomicrons. Transportation then in the lymphatic system, the chylomicrons will enter the lymphatic system and eventually reach the bloodstream, as illustrated below. Then lipoproteins, these are cholesterol transporters. Lipoproteins are tiny particles that transport fats and cholesterol in the bloodstream. There are different types of lipoproteins, and which includes 1 chylomicrons that their role is to transport dietary fats from the intestine to the liver. Then we have the very low density lipoproteins. This one's the liver, triglycerides from the liver to tissues. And then the low density lipoproteins, they deliver cholesterol from the liver to tissues. High density lipoproteins, they collect this cholesterol from the tissues and transport it back to the liver for elimination. Take note of the directions of transportation. They transfer from where to where. Yeah. Atherosclerosis, now to understand it, it's simply blockage in your arteries. And how does this occur? Atherosclerosis is a chronic disease that causes the thickening and hardening of your arteries. Yeah, so this buildup of plaques and is made up of fats, cholesterol, and other substances. For them, they can narrow down the arteries and restrict blood flow, and that's actually one of the causes of hypertension. Yeah, so once your blood vessel has been narrowed, there is a limited area or diameter that your blood will pass through, and that will actually result into the increase in blood pressure. So these lead to serious health problems like heart attack, stroke, peripheral artery disease as well. And then understanding the role of lipoproteins in this process is crucial because you can manage and prevent the complication. So atherosclerosis and lipoproteins, low density lipoproteins are often called the bad cholesterol in quotations, please. And high levels are linked to atherosclerosis. That's because they deliver cholesterol to tissues from various functions. But excess low density lipoproteins can accumulate in the artery walls, and then they become harmful, and they have an issue that they promote the plaque buildup, and they also increase the inflammation in the body. And while the low density proteins are essential for cholesterol delivery, the high levels become detrimental. Yeah, though it is good that we should have them for transportation within them and everything, but if their levels goes higher, it becomes very dangerous for us. That the excess low density lipoproteins can seep into artery walls, and when oxidized, they attract inflammatory cells, and this will lead into fatty deposits that contributes to the atherosclerosis. Lipogenesis is a process that is where the lipids are made, in which lipids are made, and it occurs in the cytoplasm, in the tissues like liver, adipose tissue, kidney, central nervous system, lactating mammary glands, all they have this process of lipogenesis occurring within them. And the major fatty acid synthesized is called palmitic acid, a 16 carbon molecule. The process of lipogenesis is actually stimulated by insulin and is inhibited by hormones, the epinephrine and glucagon. That basically involve 3 steps. That is 1, production of acetyl-CoA and the NAD pH. Then there's conversion. This NAD pH comes along with the release of carbon dioxide, which we will know also where the carbon dioxide is used in this process. Then the conversion of acetyl-CoA to malonyl-CoA, and then the reaction of fatty acid synthase enzyme complex. Yeah, let's now take note of this process and say the acetyl-CoA is the starting material for fatty acid synthesis, and therefore it's produced in the mitochondria by oxidation of pyruvate, and then there are certain amino acids and ketone bodies. Mitochondria not permeable to acetyl-CoA, so meaning acetyl-CoA cannot get out of the mitochondria. But there should be a mechanism that this thing is settled through the membrane as citrate. Yeah. So what happens here is that acetyl-CoA will condense with oxaloacetate in the mitochondria to form what we call the citrate, just like as if you're continuing with the process of Krebs cycle. And the citrate is free now. The citrate can move out of the so out of the mitochondrial matrix. So on upon moving into cytosol, here then they will cleave it by what you call the citrate layers. It will cleave it back and form our acetyl-CoA and oxaloacetate. Then what happens is that the acetyl-CoA is ready to go with the process, but then we don't have yet the oxygen. Oxaloacetate, carbon dioxide, we don't have yet the carbon dioxide. We don't need oxygen in this case. And then oxaloacetate in the cytosol is then converted to malate, and then that's done by malate dehydrogenase will convert the malate to pyruvate with production of NADPH and carbon dioxide, and the NADPH is what we said was needed also. Produce acetyl-CoA with NADPH and then release of carbon dioxide. And now what happens is that the transport of acetyl-CoA from the mitochondria to cytosol is coupled with production of NADPH and carbon dioxide. Both of them are utilized in the synthesis, as we will see again later. And this is the pyruvate citrate malt shuttle. That pyruvate is converted to acetyl-CoA and then to citrate. Citrate is moved out, and then you find out how it is going into malate and NADPH and everything. And then the formation of malonyl-CoA, this is the first step of fatty acid synthesis. The acetyl-CoA is carboxylated, that's one, and that meaning I can say is activated to form malonyl-CoA. So this reaction is regulated by acetyl-CoA carboxylase enzyme. So the reaction is ATP dependent. It requires ATP for it to take place, and requires biotin 2 for carbon dioxide fixation. That's where we use our carbon dioxide that we had produced. That acetyl-CoA carboxylase is also planning that on that very day. As we fix it, as we fix the carbon dioxide also. There's need for biotin as a cofactor for this whole process to go. I remember we've talked about micronutrients, as B vitamins. Most of them generally are involved in energy metabolism, which is now here that we have biotin, which is vitamin B7, is involved in this process of conversion of acetyl-CoA to malonyl-CoA. Yeah, so you get to understand the relationship and the function. If you say B vitamins are very important for macronutrients metabolism, you get to understand it from that perspective. And a fatty acid synthase enzyme is a multifunctional enzyme system with 7 different enzyme activities and acyl carrier protein. Yeah, what happened that we've been discussing that. It adds this process will add 2 carbons every cycle through addition of malonyl-CoA and loss of carbon dioxide. What's the advantage of fatty acid synthase complex here? It's that there is one gene can code for all enzymes in the equal concentrations. So there's a process also, the process enhancement and therefore efficient. It's also intermediates of the reaction can easily interact with the active site of the various enzymes. Also known the first complex, the first domain, is also known as the condensing unit. It is the initial substrate binding site that contains the enzyme beta-ketoacyl synthase, and that is a condensing enzyme. and then acyl transferase AT and malonyl transacylase. Second domain is also known as the reduction unit. It contains enzymes, dehydratase, enol reductase, beta ketoacyl reductase, and acyl carrier proteins. The ACP is a polypeptide chain with a phosphopantothene group, to which acyl groups are attached in the thioester leakage. And we have a long chain of these polypeptides. So it's where these molecules are attached to everything. And the third domain here contains the esterase. There's an enzyme that breaks the ester bond between them, and it is used for the synthesized fatty acids. The major fatty acid synthesized is palmitic acid, like we mentioned earlier. And then let's look into the reaction of these things. That a 2-carbon fragment of acetyl-CoA is transferred to an acyl carrier protein, which is, let me say here that this is a cysteine. The structure of this is right here, that we have a cysteine residue and an ACP here. So what happened is that an acyl-CoA molecule has been transferred here and there on the ACP. Yeah, so that is the first step there. And then the enzyme malonyl-CoA ACP transacylase here transferred the malonate from malonyl-CoA to bind to the empty site of what? ACP. So the acetyl unit attached to the cysteine is then transferred to the malonyl group, and this moiety loses carbon dioxide, which was added by acetyl-CoA, and then decarboxylation is characterized by beta ketoacyl, catalyzed by the beta ketoacyl ACP synthase. Carbon dioxide is never incorporated into the fatty acid carbon chain as well. And decarboxylation, sorry, those are just the visuals. Decarboxylation is accompanied by loss of free energy, allowing the reaction to proceed. Looking here that one, we look at this molecule and we are adding malonyl-CoA. What happened is that we lose the CoA here, and then we add our malonyl to ACP protein carrier. Yeah, so that is basically what it is, or what the reaction really means. So the enzyme beta ketoacyl ACP reductase reduces the ketoacyl group into hydroxyacyl group. Then the reducing equivalents are from NAD. So beta hydroxyacyl ACP undergoes dehydration, and the molecules of water is eliminated to form a double bond. Between the alpha and beta carbons. Now the second NAD here, the second NAD here is dependent reduction, catalyzed by the enol ACP reductase and it yield an acyl ACP. The 4-carbon attached to ACP is butyryl group, just like you've seen here. That is the other step there, and then we have the transacyl trans-enol ACP. Then you put the reductase, and then you want butyryl ACP with the 4 carbons, and then this butyryl chain is attached to ACP, is then transferred to the cysteine residue. So it moves from ACP, it is from ACP to move to cysteine residue, so that the ACP can now accept another 2 carbon of malonyl. At the end of all these cycles and the fatty acid synthesis is complete, and then you get 16 carbon fully saturated fatty acid. So palmitoyl thioesterase then separate the palmitate from the synthesis completing the process. So once the palmitate is been separated from the synthesis, then the system is complete. Fatty acid elongation occurs in the mitochondria or endoplasmic reticulum by a separate mechanism that is catalyzed by elongase enzyme. And this process is a successive addition of acetyl CoA molecule to lengthen the chain. So basically, dear learners, I can give you all that as the final thing I can tell you about this lecture, and I want to say thank you very much for attending.